Good morning, everybody. I am Kim Nye, the president and founder of TESS Research Foundation, and we are really excited to bring our families another webinar. This morning, we will be talking with Alleigh Boyd, who is a licensed certified genetic counselor currently working at Stanford Fertility. As always, these are educational webinars. We're not trying to give medical advice, so please talk to your doctor or healthcare provider about specifics for your family. Having said that, thank you for joining us today. Alleigh, do you want to say hello, and then we'll get on with your presentation?
Yeah, hi, thanks for having me. Yeah, this is definitely not supposed to be fully comprehensive. Each of these options are going to have a lot of different considerations. So just to give you kind of a general overview of what maybe reproductive options are out there. So I'm gonna go ahead and share my slides. So just kind of basic overview. There's another amazing webinar that you guys put on where Ellie goes over kind of genetics 101. So I'm not gonna spend a lot of time on genetics or inheritance patterns or anything like that, but just a super quick refresher. When we're talking about these kinds of genetic tests, the context of this is that we're made of billions of cells. In our cells, we have our chromosomes, which is just the package of information, how we pass on all of our genetic information to our children. And then if you were to actually unwind that chromosome and look at a specific sequence of DNA that performs a function, like SLC13A5, you're talking about kind of a specific region that then encodes a specific protein. So for this particular condition, kind of the focus of this talk, we're really going to focus on autosomal recessive inheritance, in that there is this 25% chance or 1 in 4 chance that there could be an affected child with each pregnancy. So that means that couples could have multiple affected children. And this is really assuming that carrier status is confirmed for both of the parents, and that would then infer that 1 in 4 risk. The other thing to keep in mind is that for the genetic testing options for reproductive planning, this carrier status is also very likely going to need to be confirmed before you can consider these reproductive options, some of them. But the overall takeaway is that you actually have lots of options for family planning. So we're going to go through a bunch of these. So you could have preimplantation genetic testing for a monogenic disorder. You could have prenatal diagnosis, so during pregnancy. You could wait until the baby is born and confirm status after birth. You could also consider other options in terms of gamete donors, like sperm or egg donors, and then also adoption as a reproductive option. The huge caveat to this and the whole talk is that although there are lots of options, each of these options may not be a good fit for each individual or couple based on maybe their religious or ethical considerations or even geographical differences. Not all of these options may be available state to state or country to country. So the first thing I want to talk about, and I really also have to say that since I work in a fertility clinic, this is my bias. I know a lot more about this option than basically any of the other options, but we'll kind of go over them very, very broadly. So preimplantation genetic testing is an option that happens within an IVF cycle. IVF, or in vitro fertilization, is a process where a woman's natural menstrual cycle, is manipulated through fertility medications. These fertility medications are hormones that your body typically makes, but just in mega doses. And so instead of growing 1 egg that cycle, we're trying to increase the number of eggs that grow during that month that then can be used to make embryos. The other thing to just be aware of is that although we are trying to grow more eggs, it actually doesn't take away from your future fertility. So you have a set number of eggs that will just naturally die off during that cycle, probably about 1,000 eggs a month, versus in your natural cycle, you're really only using 1. So your body kind of devotes all of the energy and resources to that 1 egg, you ovulate that 1 egg, and the other 999 just die off. But you may have 10, 15, 20 other eggs that could be harvested or used or maximized during an IVF cycle that, again, would otherwise just die off. So again, not taking away from your future fertility, just maximizing that month's potential. The first thing certainly you would need to do is you need to meet with a reproductive endocrinologist. These are the physicians in a fertility clinic. And if possible, not all fertility clinics have genetic counselors, but if they have one, then certainly meeting with a genetic counselor to talk about the IVF process and PGT in general would give you a good idea of what that process will look like for you. And again, like I mentioned, you have to know your carrier status. And not only do you have to know that you are a carrier, you have to know the exact mutation that both you and your partner carry in order for this option to work. So preimplantation genetic testing is the process of actually testing embryos for specific mutations prior to implantation, meaning prior to pregnancy. So this requires the IVF, and it also requires an embryo biopsy. So the process kind of depicted here is that, you know, as we're giving you the medication, we're hopefully growing multiple follicles, then you would undergo an egg retrieval. You're usually asleep when this happens, where they then aspirate or take the eggs out of the follicles, and then those eggs would be given to the embryology lab. So when we are trying to make embryos, you then have the egg. When you do pre-implantation genetic testing for a monogenic disorder, you actually always have to do ICSI, where you are physically injecting a sperm inside of the egg. The reason why you do that when you are testing for a single gene disorder is really just contamination. We don't want extra sperm to contaminate the sample because certainly sperm has DNA in it, and we don't want any extra cells that are coming out with the egg to contaminate the sample. So we want to make sure that what we are testing when we test the embryo is really representative of that original egg and original sperm. So then you would grow that embryo out until about day 5, maybe day 6, maybe day 7, in which case you have this embryo that is about 100, 120 cells, and you actually have 2 very distinct cell types at this point. You have inner cell mass, which is a cluster of cells, which would turn into the fetus if this embryo created a pregnancy. And then you actually have this other outer layer of cells, what we call the trophectoderm, that turns into the future placenta. So when you take a biopsy from an embryo, what you're doing is you're actually taking 3 to 5 of the future placental cells. You're sending that sample off to the genetic testing company, and then the company would tell you what the results are. So in this case, they would, in most cases, be able to distinguish between the affected state, the carrier state, and the unaffected state. So you actually would have the option of either maybe transferring an unaffected or a carrier embryo. So then that transfer process is actually kind of the less complex part, but obviously the most important part in terms of getting pregnant. So it's similar to maybe a pap smear where they are, going through the vagina, but they actually go through the cervix with this catheter, and then they would release the embryo into the uterus. When we are doing this process and waiting for the results to come back, these embryos are actually frozen. So we freeze the embryos, wait for the results to come back, and then you would decide based on the results which embryos to transfer. And so since the embryos are frozen, and a lot of times, although not always, there are multiple embryos in that cycle, there is the potential that one cycle or a couple of IVF cycles may yield enough embryos for multiple pregnancies. So you could then go back and use these embryos if you were considering a future pregnancy. There are definitely good things and bad things about this option. So the biggest thing that you should just be aware of is that there is a relatively long setup period before you would even go through IVF. So the time when, like if you were meeting with me today to talk about PGTM, there's probably about a 6-month lag between that consultation and when you could potentially do a transfer of an embryo. And so if this is an option that you're considering, I would strongly recommend you give yourself a good buffer of time just to not further delay pregnancy, because we don't know what's going to happen in the IVF process. We don't know how many cycles it might take. So just to be aware of that. The other thing to be aware of is that this often requires other family members to be involved. An embryo sample, that 3 to 5 cells, is really just so much smaller than any other biological sample, like a blood sample or a saliva sample, that there are big limitations to finding single gene mutations in that sample. And so they often need another reference. So this could be for a couple that has an affected child. This may be just providing a DNA sample from your child, which is usually a buccal swab, not a blood sample. Or this may be the potential grandparents, just depends on the family. But just, again, something to be aware of is that this may not be as private of a reproductive option as others might be. It does require IVF, and it may require multiple IVF cycles. IVF is no guarantee. Even if you've had pregnancies previously, that doesn't always guarantee that you're going to have a good outcome or multiple embryos at the end of IVF. IVF is very expensive, depending on your coverage, depending on your state, depending on your financial situation. This may or may not be a good option for you. Medical insurance does often cover PGTM, the actual genetic testing portion, but it may or may not cover the IVF. And the IVF is really is the significant financial burden in this situation. And like I mentioned, it doesn't guarantee a healthy child, either because you could not have an unaffected embryo or a carrier embryo, but also that there are many other things that factor into health. And so, another child could be born and they could have some other different medical issues. So just to be aware of that. And we really don't know what the impact, the kind of the long-term impact of IVF or embryo biopsy is on a child. There's no non-invasive option currently to do a biopsy or to do testing of the embryo. There are certainly things that are coming down the pipeline in terms of maybe potentially non-invasive options, but those don't exist yet. They're not clinically available yet. And again, like I mentioned on the previous slide, is that this may just not be an option for religious or ethical reasons. There may be an issue with producing multiple embryos at once, or using assisted reproduction in general. It just may not be an option. So the alternative to this, or one of the alternatives to this, is prenatal diagnosis. So if you were to just get pregnant on your own, or even if you were to use assisted reproduction, but you didn't do any sort of testing on the embryo prior, then this is the way that you could find out the information during a pregnancy. So typically you are talking about doing one of two procedures, a chorionic villus sampling or an amniocentesis. So chorionic villus sampling, or often people will just call this a CVS. This is performed at 10 to 12 weeks in pregnancy. In this picture, this is showing a transcervical CVS, where the biopsy pipette is being threaded through the cervix, but there is actually a transabdominal through the stomach option as well. This is an ultrasound. This is this fetus, just to orient you. This is the placenta. And what the biopsy is doing in this case, or what the CVS is taking a sample of, is of the placenta. So it's using that as a representation of the fetal genetics, which is true because the placenta, like we were just talking about, comes from the same embryo. Versus an amniocentesis. So this actually happens a little bit later in pregnancy. This is about 15 to 20 weeks. So you're further along at this point. Similar sort of procedure, or perceived procedure. So this is transabdominal, ultrasound probe. This is a needle, but as opposed to taking a part of the placenta, what they're taking is some of this amniotic fluid. So neither of these procedures are actually taking a sample from the fetus itself. It's using other sample types as a representation of the fetal DNA. And then they can take that amniotic fluid, they can culture those cells, and then they can test them for the specific mutations that you're looking for. The benefits of these are that these are diagnostic. You're going to get an answer that's 99.9% accurate. No test is 100%. But the downside of these procedures is that they are invasive. They require, a needle to penetrate your skin, and theoretically the amniotic sac or the placenta, and that there is a risk of infection with these procedures, like anytime you put a needle in your skin, and there is a risk of miscarriage as well. That risk really depends on the clinic, and it also likely depends on the clinician performing the procedure. General risk estimate is maybe like 1 in 1,000 for miscarriage. But other centers might quote a more conservative risk of maybe 1 in 300 to 1 in 500. In any of those cases, it's less than 1%, but just something to be aware of, that there is this potential risk. The other kind of consideration here is that you're already pregnant. And so, you know, is the information that you get from this testing going to be helpful or not? That might be something that you may consider in doing this testing, meaning, you know, are you going to use this information to discontinue the pregnancy or to have a termination of the pregnancy? Then this information may be helpful. If you just want it for reassurance, again, that's also helpful. But if you feel like you wouldn't change your management in any sort of way, then you also could consider just, you know, neonatal testing or testing after birth as opposed to one of these invasive procedures. Similar to the non-invasive technique on the embryo that may be coming down the pipeline, there is this future option of NIPT in pregnancy as well. And so because the placenta comes from the same embryo that made the fetus, the placenta, the cells that are kind of on the outside of the placenta that are attached to the uterus, as those cells die off, just like any cell will do, has a natural, you know, cell cycle, the DNA from those cells are actually released into the mom's bloodstream, or the carrier of that pregnancy's bloodstream. And so there is current testing that's looking at chromosome abnormalities using this method, like Down syndrome. But the single gene part or potential isn't really quite ready for clinical use. You might have to check. There are some options for maybe more common single gene disorders. But again, this might be an option in the future of doing this non-invasive version. So the neonatal testing, I think, is going to be the most straightforward, is really just waiting till after birth to perform testing. This is usually going to be done on a blood sample. This is usually cheaper and faster than doing the more comprehensive testing that was done originally to find the diagnosis, because you're going to pinpoint just those specific mutations. But the downside, obviously, is that the baby is already there, and so you might not have as much flexibility in terms of what you do with that information, although helpful for, you know, management. Other potential options are gamete donors, sperm or egg donors. So a gamete donor in general is someone who provides sperm or egg to another individual or couple outside of a romantic relationship. This could be a known donor. This could be a family member or a friend, or it could be an anonymous donor, someone who donated to a donor bank or as part of a donor agency. Usually these individuals are young and healthy, versus the known donor. There's not as much restrictions because there is that relationship, and that relationship may be more important to you than other medical considerations. In either of these cases, the benefit would only be if this gamete donor was not also a carrier for a mutation in SLC13A5. But the assumption is that they're not a carrier, and if they are not a carrier, then that's going to significantly reduce the chance of having an affected child. So you're essentially replacing one of, you know, the parent's biological material, which would then mean that the risk to potential offspring in this case is really a 50-50 chance of being a carrier. So they would still have that chance of inheriting a mutation from the biological parent, the intended parent, or not, but you're kind of getting rid of those other two possibilities of the affected child or the carrier from the other biological. And some couples may like this option because it still gives the opportunity for one of the intended parents to be biologically related to the child. There's also the potential to make embryos using both egg donor and sperm donor. This is a more rare option, but the benefit here of using a sperm donor or an egg donor or both is that you have a little bit more control over who that donor is. Meaning, again, like you're choosing someone you know, or at least for the anonymous donors, you have potentially a lot of donors to sort through, and you're choosing one that feels the best to you. So again, you have a little bit more control there. Sperm donors specifically are typically less than 40 years old. Usually they're in their 20s, like both egg and sperm donors are typically in their 20s. This option is potentially the least expensive option because you could have the option of doing at-home inseminations as opposed to going through a clinic where you have to then pay for medical fees and all of those associated visits. You do also have the option of doing in-clinic inseminations, or you could do IVF if there are other considerations, or if you really wanted to shorten the timeline and you wanted to maximize the number of eggs that you were using or something, you do have the option of doing any of these. But again, sperm donor is potentially the least expensive option because you do maybe have that at-home insemination option. Egg donors are also a good option, but they're a little bit more complex in terms of the process. When you use an egg donor, that automatically means you're going to be using assisted reproduction. These are women, so anonymous donors are usually younger than 35. 35 is kind of the cutoff in terms of this arbitrary advanced maternal age. So usually they're younger than 35. Again, usually they're in their 20s. But either with sperm and egg donors, especially the anonymous, forms, is that you may have limited personal or family medical history. So you might not know exactly what's going on in the family. All of that information is by report. And so it's really just, trusting that the donor is both reporting all the information and that they know all of the information that's going on in the family. And that because this is a rare disorder for both an anonymous, actually for really any donor, you would likely need to pay for carrier screening for SLC13A5 just to confirm that that person is not a carrier. And that can be expensive depending on how you're doing it and who the donor is. Egg donor option is more expensive than sperm donor because you're either paying for the eggs, you're either paying for frozen eggs from an egg bank, and again, these are like anonymous. This is the anonymous option, the frozen eggs, and that can be a good chunk of money. But then you also then have to take those eggs, you have to thaw them, you have to inseminate them, either you're using your partner's sperm or a sperm donor's sperm, and culture them and then transfer them. So this is going to look a little bit like IVF, except for you're not actually doing the retrieval of the eggs yourself. And then you also have the option of a fresh cycle, a fresh donor cycle, where the intended parents are responsible for the IVF and the other medical costs for the donor, travel, testing, required consultations. But this option often yields many more eggs than a frozen cycle, because donors are typically young and healthy and have a high egg reserve. So if you're considering multiple children in the future, then a fresh cycle might be a good option. If you're really just wanting one more child, then maybe the frozen egg option is good. Again, with the known donors, these become a little bit more complex because it depends on like who the person is and how old they are and what their fertility status is in terms of like the success rate or the yield of that person. The other option is adoption. And there's actually two things in terms of adoption that I want to talk about. So there is actually embryo adoption. So embryo adoption or donation, you'll kind of see those terms used interchangeably. They technically mean different things, but this is when, as opposed to you choosing the sperm donor and choosing the egg donor and then making embryos, this is a situation where you are accepting embryos that were donated by another couple. So this may be a couple who went through IVF themselves and have excess embryos, but they've completed their family and they no longer have use for these embryos, but they don't want to discard them. So they may donate them to a donation or an adoption program. this is the similar limitations in terms of like anonymous sperm or anonymous egg donors, in that you may have limited information about the egg and the sperm provider, and you also have less control over maybe who those people are. But this actually may be cheaper than other options. So this may be cheaper than like the egg donor option, but it might be more expensive than the sperm donor option. Adoption in general, where you're using both a different sperm and egg provider, whether that's you're choosing those people or you're accepting an embryo that's been donated, may actually take away the potential emotional burden of only one of the parents being biologically related. This may not be an issue for some couples, and some couples it might be an issue. So this might be a good option if they just, you know, want to take that factor out of the equation. And that this option, just like using, again, like the egg donor or sperm donor option, this gives the intended mother the option of actually carrying the pregnancy, which is something that a lot of people really want. And then the other adoption option is the more traditional adoption that we think of, where it's actually a live-born child. And this could be a domestic adoption within your own country or an international adoption from another country. But adoption, just like assisted reproduction, can be very expensive, and the time frame is often unpredictable. And you may or may not have much information about the biological parents of that child. So benefits, limitations to all of these things. So the conclusion is that there are lots of reproductive options after a genetic diagnosis is made, but just like Kim said at the very beginning, consultation with a genetic counselor or a physician is really where you should start, because again, not all of these options may be right for you just based on how you feel, or religious or ethical considerations, or it may not be an option just based on like where you live. So consulting with someone that knows what the options are that are available can really help you figure out which option is right for you to build your family. So with that, I think I'll either open it up to questions or hand it back over to Kim.
Thank you so much, Alleigh. That was really great. I learned a lot, honestly. And what we're going to do is, well, I guess my two big impressions would be, this is a newly discovered disease, so it's amazing that we have these options of testing, before birth or even before an embryo is implanted. But I also am always just struck with how much our families have to go through. Like this is a topic that, other families don't even have to consider. So my heart goes out to all of us that are, even having to complicate or contemplate these conversations and these decisions. What we're going to do for the Q&A is I'm actually going to stop recording this session so that the people who are participating live can answer or ask and have answered the questions that are important to them. So I'm going to push stop on the recording.